So in this final set of lectures for the PH 601 course in epidemiology, I want to talk about genetic and molecular epidemiology. These are very important areas of epidemiology. There are specific tracks in public health specific for genetic or molecular epidemiology. And I would say probably one of the most interesting fields of epidemiology. That's just my personal opinion. But the reason I think that is because with genetics and our ability now to look at data at a molecular level, we are able to look at many other variants that are potentially related to diseases that we did not know about before or we were unable to look at before. You may have heard of personalized medicine and how that's becoming kind of a hot topic or has become a hot topic in the past several years. And the idea there is we would be able to hopefully in the future be able to determine the right course of treatment for someone with a particular disease given their genetic and molecular makeup. Now, that's a slightly tangential point to this lecture, but nevertheless, with genetic epidemiology, our goal is to look at the relationships between genes and diseases. So for me, I think of genetic epidemiology as a beautiful pairing or marriage between epidemiology and genetics. It's two separate fields, as you know, but the pooling of the two makes up this neat field in itself. So we now know after several weeks in this course that epidemiology is really the study of the distribution, the determinants, how to control those determinants of health-related states and events in populations. Now, genetic epidemiology takes genetics and combines it with epidemiology to create a discipline that's closely aligned to epidemiology, but it has a focus on the familial and in particular the genetic determinants of diseases and the joint effects of genes and non-genetic determinants on the relationship with those diseases. So the gene-gene relationships and the gene-environment relationships. There is a paper by Burton and colleagues in The Lancet in 2005 and is also provided in the Blackboard reading. This is a nice paper that summarizes the key concepts of genetic epidemiology. And I really recommend that you read this closely because it provides you with a nice introduction to this field. And as I introduced in the lecture, this is a field that's certainly growing and will become much more used and applied as we look at relationships between determinants and diseases. So more specifically, genetic epidemiology is an epidemiologic evaluation of the role of inherited causes of diseases in families as well as in populations, but it usually begins at the family level, and I'll talk about that in this set of lectures, with the goal in mind to describe the inheritance pattern of a specific disease. If possible, genetic epidemiology seeks to identify the gene and the marker on the gene itself that is associated with that disease or with the susceptibility of acquiring that disease or presenting that disease. It takes into account the relationships between genes as well as the relationships between genes and the environment, and that's important because as was discussed in our book and clearly something that you have to think about as you explore patterns of diseases is that there is rarely a situation where a particular disease is due to a specific gene alone. And examples exist for that. We can look at birth defects and other things for which the environment is also having a role or plays a relationship with that gene expression. And so, as described by Morton in 1982, which was well before our ability to look at the genetic makeup of an individual, he basically described genetic epidemiology as a science which deals with etiology, the distribution and the control of diseases in groups of relatives and with inherited causes of diseases in populations. So again, genetic epidemiology, pairing genes, and epidemiology, which is looking at patterns of diseases in populations. Now, I just want to briefly summarize the gene itself and what we're talking about, and I apologize up front if this is really elementary for some of you. but I just want to make sure that everyone is familiar with some of the terms that I'm using. So this is the anatomy of the cell. This is the human cell, and we are eukaryotes in definition. And the key message I want to show here is the nucleus, which is this rounded area here. In the nucleus is where we find the chromosomes which house the genes that we're going to be discussing in this set of lectures. The nucleus itself is called the powerhouse of the cell, and that is because it's the site of where the chromosomes are made and for the most part where the chromosomes are located. Now, humans have 46 chromosomes, 22 of which are what's called autosomal, and one pair are sex chromosomes. And as you know, we have two types of sexes, males and females, and the sex chromosome pairs differ. For females, it's an XX set of chromosomes, and for male sex, it's an XY. Now, each pair of autosomal chromosomes is considered homologous, or the same, and that is that they contain the same genes ordered in the same way. And each member of that pair, one comes from the mother and one comes from the father. Now, chromosomes comprise deoxynucleic acid, or DNA, and we now know that the human genome contains over 3 billion base pairs of DNA, of which about 1% encode for proteins. Human genes are located on chromosomes. That's true for all genes, by the way. And that we now know, given the results of the Human Genome Project, which was geared at fully sequencing the human gene, the human chromosome, which was completed in 2003, that the human genome comprises 22,000 genes. This is just provides you with a human karyogram. A karyogram is an illustrative representation of an organism's chromosomes. And once again, you see that we have, as humans, there are 22 chromosomes that are autosomal. They have two pairs, or a set of pairs, I should say. And then we have the X and Y sex chromosomes. And while we are an advanced species, there are other species of animals that have more chromosomes than 22. Several examples of that exist. The horse has, I think, around 46 chromosomes. And if you look up, there are some organisms that have up to 100 chromosomes. Now, I want to define two key terms that I will use throughout the lecture. The first is genotype. This is essentially the overall genetic constitution of an individual. And genetic epidemiologists generally refer to a particular location along the chromosome using this numbering and lettering system shown here. The phenotype, in contrast, is the measurable characteristic of the individual. It is the trait. It is that genetic expression that is presented at the individual level. Now, this is essentially to describe the genetic etiology of a disease in its simplest form. And you can also apply this kind of logic to genetic expressions that don't necessarily mean or confer disease. But nevertheless, what we're talking about is we first begin with some genetic variant or variation in the gene, which leads to an expression on that gene, and then that expression confers some particular product, be it a protein or something else, and then that lends to some alteration in the physiology for that characteristic, which then presents in the physical manifestation or the phenotype. Now, there are a myriad of different kinds of molecular markers for diseases, and I'm just going to walk you through some of the key categories just so that these terms are familiar for you. Now, first, I want to define what I mean by molecular marker. This is a measurable, that's key, indicator measured at molecular level by definition for some biological state or condition. And now, measurability is key. If we believe that there's a marker, but we cannot assay it, then again, we cannot do molecular typing, right? But nonetheless, we're talking about here in these next two slides, markers that we are able to measure, okay? So we have a first category of a molecular marker I want to discuss is the genomic. These are genes or DNA sequences with known physical locations on the chromosome that we use to determine if individuals, or sometimes even species, differ. So an example would be a tumor suppression gene located on a known loci of the chromosome. We also can talk about the breast cancer 1 and 2 genes, which can mutate, and then that confers an increased risk of breast cancer. There are things called SNPs, or single nucleotide polymorphisms, and these are small regions on our DNA that vary from person to person. And then you can take that even a little further and talk about what's called microsatellites. And these are essentially tandem repeats in DNA sequences that which are variable within the alleles of an individual. We won't talk about microsatellites anymore in this particular set of lectures. It's just provided as an example for you. The next type of molecular marker would be the metabolic or metabolomic marker. This is a marker that basically is some sort of molecular metabolite that's generated from some cellular level process. So you can have markers that are intermediates of a metabolic process. You can have a hormone, that's a metabolic marker. You can have a marker that's basically some sort of molecule that's part of the signaling process of the metabolic process. So lots of examples here. We think of metabolic markers. We can talk about lipids, sugars, amino acids, etc. To put that into something that's applicable that we all are familiar with, you can think of coronary heart disease and the metabolic markers of HDL and LDL, which are those high and low density lipoproteins. And there's a neat paper that I reference here for you if you're interested in a little more about understanding metabolic markers and how they are used to predict risks. This is specific to the risk of cardiovascular disease. And again, just an interesting paper if you would like to dive into that a little more. And then the other category I want to talk about is the proteomic marker. And as the term implies, it's looking at markers associated with proteins or protein expression patterns. So there are a myriad of these kind of biomarkers, and I'm actually listing here on this table some well-established proteomic markers and what their clinical use is and what cancer they're associated with and how they're captured or collected. So we can talk about, say, prostate cancer. The biomarker that is known to be associated with that is the Pro2-PSA. It's used clinically to discriminate cancer from benign disease, and it's collected in this serum. We can talk about, say, testicular cancer, and we know that the biomarker of alpha-fetoprotein can be used to help manage cancer treatment for testicular cancer, and this biomarker is measured in both serum and plasma. But again, this isn't an exhaustive list by any means of proteomic markers, but just a few that are pretty well understood and how they're used clinically. And I think that's important to just see this to get kind of a sense as to how do we move from the molecular level to the clinical level.